Awesome. I'm Austin. I'll be presenting with Jan about how to prepare for novel incursions using sexy genes to call lineages in the myrtle rust pathogen.
Hi, my name is Zhenyan, and today I will present this talk with Austin. We are for Zhenyan lab. So first, we wanted to thank people who involved in this project, and especially thanks Benjamin, Thais, and Perry. We know myrtle rust pathogen can infect different hosts, and different strands of the myrtle rust fungi can cause similar symptoms on different hosts. Thanks Thais for providing these nice pictures. And here we can see this Zhenyan isolate, which infect the guava, CA infect the kumquat, and Zhenyan on rose apple. These are different strands, but they have similar symptoms on infected hosts. So we are looking for a method which can help us to distinguish different isolates. Then we're thinking about using the mating type genes. So before talking about the mating type genes, we know the sex of mammals are determined by sex chromosomes. For rust fungi, they don't have sex chromosomes, they have mating type instead. The mating type is determined by the mating type genes. And by mating type locus, there are two mating type locus in rust fungi, the PR locus and the HD locus. The PR locus consists of at least one pheromone peptide and a pheromone receptor encoding genes. And each HD locus consists of two HD genes, or BW1HD and BE2HD. To be able to have mating compatibility, both these two locus needs to be different from each other. And we know rust spores has two nuclei with different mating types. In previous study, we know the PR locus in rust fungi, cereal rust fungi, as well as the Apicidia are likely to be biallelic. But the HD locus are multi-allelic, and HD genes in the HD locus can be very diverse. So we are thinking about whether we can make use of the variations in the HD genes to distinguish isolates. This is how the HD genes looks like in the myrtle rust fungus. We use the BW1HD of the pandemic strain haplotype A as the reference, and we map the BW1HD alleles from Brazilian strains and also the South African strains to the pandemic strain. We can find there's lots of snippets, and there's lots of variations in the BW1HD1. And for the another genes in the HD locus, the BE2HD, we also found these genes are highly variable in the myrtle rust fungi. So we have this. So we will base on this to develop our methods to identify isolates.
Cool. So now that we understand a bit more about the biology of this region, I'm going to step you through some of the workflow that we've been developing. To start off, we did a PCR and we developed primers that amplified this HD region. We then did a gel electrophoresis to do a quality control to make sure we've amplified the right area. We then performed long read sequencing with nanopore to produce reads of this region. Using these reads, we can then reconstruct the HD loci. Once we have the HD loci reconstructed, we're able to perform a phylogenetic study to compare to other isolates and create a gene tree. And with this gene tree, it allows us to diagnose whether this is a new isolate and infer its origin. So just the workflow there. The first step of this was designing the primers. So as Jan's already explained, this is a highly variable region, this HD region. So we had to find more conserved regions either side of both genes to develop these primers. We managed to find some for the BW1 gene, a pair for the BE2 gene, and then we could combine these to develop one for the full HD loci. To ensure amplification of non-pandemic strains, we also had to develop degenerate primers. So what that means is it's a mix of oligonucleotide sequences that have different possible bases at different sections. That's because even in these conserved regions of the HD loci, there's still variation between different biotypes. Once we had our primers designed, we then wanted to look at how specific they are towards apisidae. So we amplified the myrtle rust extracts we had, as well as a number of other fungi and close related species of other rusts. As you can see here, we had good amplification after 35 rounds of PCR, but no amplification at all for any of the other fungi. This is really important, especially when it comes to looking at using mixed samples or environmental DNA. We don't want to be amplifying other species that we're not looking at. Now that we are confident that we can amplify this region, we wanted to use and see how broadly applicable it is to those other exotic types. So as you've heard from Taze earlier yesterday, around the globe, there's a pandemic biotype, which is common here in Australia, but in South America, there are multiple biotypes. So as these different biotypes all have very variable HD regions, we wanted to make sure we could still amplify these, and so we could see if we can use this as a way to detect these different variants. So we were able to successfully amplify 14 different isolates that were living across 11 different host species for all across South America. We managed to amplify all of these successfully with the degenerate primers. So now that we'd amplified them, we did nanopore sequencing with them. And this is kind of one of the outputs we got. So if you can see here, this is the reads mapped to the reference genome. So all the bits with grey are bits that match perfectly to the reference genome. So you can see for this example, the BW1 HD gene has a lot of variation. All those coloured bars are different SNPs compared to the reference genome for this sample, while the BE2 HD gene looks fairly similar. We then continued this for all of the samples, and we're able to see that for every single isolate that we sequenced, at least one of these genes did have significant variation that we could identify to call which variant it was. So that's just it there. So now that we've done that, I'm just going to walk you through. So far on the development of this workflow, we've identified this highly variable region. We designed degenerate primers to amplify this HD loci. We checked and made sure that it was amplifying only apisidae. And we tested these on exotic strains and different variants. So now what's next is seeing how it can be applied in the biosecurity setting for variant detection and diagnosis.
And this is a case which we use in try to diagnose the Astrazeneca apisidae in real world. First, this is the positive control. We amplifying the HD locals in the pandemic strains and map to the itself, the reference genome. And the gray block in the graph is the coverage, and we didn't find any snippers. So yeah, the positive control is assessed. Then we have some samples for some spores, essential samples for study. And we also do the same thing, is map to the reference genome. And we also can't find any snippers. So this is likely that it's the same strands as the pandemic strands. And we also have got some samples from Western Australia. And also we can't find any variations in the HD locals when comparing to the pandemic strain. Besides, we also tried with using the primers to amplify the HD locals from leaf extractions and also the air samples extraction. And we can see in these two cases, we can also amplify this HD locals successful, and the nanopore long reads can also map to the pandemic strain well. So that's all today. To sum up, we proved the possibility to use the HD lockers to design primers to diagnose the Estrepsina faciei isolates. And because the HD lockers are highly different between the nucleotides, so this method is also have high nucleotide resolution. And because the HD lockers is also can also be used in the other rust fungi. So by designing the primers based on different genome of different rust fungi, can also use this method. So it's a very highly adaptable workflow. This project is still in progress, and in the future, we will test these methods on mixed DNA samples to see whether we can successfully rebuild the alleles for each isolate. And then now we are working with people from South America, so we will be able to use more genome resource to continue to improve and finalize the primers to make them available for more cases. At last, we hope to work with Louis Su to develop PCR-based HD assay. Yeah. Thank you so much for listening.
Similar question to what I asked Benjamin. What do you think the error rates are like in terms of false positives, false negatives going after those loci? In particular, given that we really don't have really good sequence from a lot of isolates around the world of those regions.
I think there's no transposon elements inside HD locals. I'm sorry. Could you repeat your questions?
So yes, I think from what I understand is you're asking how we can make sure we can amplify those rare strains. Is that what you mean?
Diagnostics is about differentiation. So, a false positive is saying you believe it's this and it's not, and a false negative is saying I don't think it's that and it is. So they're really important criteria for the development of a diagnostic. And right now there's only one genome that's really got a lot of definition to it. The one that Perry developed. The other ones are very patchy. Do you think? Can you give it a feel for how good this region is going to be, given that the paucity of data around sequences from other Australopexinias from around the world? That's the guts of the question.
So I think one critical part about it is it's able to tell us if it is a new isolate. If we are able to detect that it's a new isolate, because we saw how variable this region is, it's not likely we'll find many that match and get misclassified, and there's not many false positives in that sense. There are the chances we haven't sequenced them yet, but we'll be able to identify that it is a new strain or a new isolate.
Any other questions?
Yeah, I mean, I think, sorry, I was just going to say that we do actually have more strain data as well, so we can validate those things. Better examples, that was what Tejas was presenting on the genomes